we will define CpG islands, and we will talk about the methods. But the very first thing that I want to mention here is why in the world this is of importance. Why do we want to study this? And there are several reasons, but one of the reasons is that in 2001, the human genome was sequenced. So that was a big deal. You know, Science and Nature both led with this story. I mean, imagine if they didn't. So, you know, big deal. And so everybody was very happy, deservedly so. And at that time, people were already talking about certain things which I didn't understand, because I was hearing words that went like, OK, now we have the genome, but we don't know how to read it. It's like, what do you mean if you have the genome? I mean, this didn't make much sense to me until, you know, I kind of realized that, you know, after we've sequenced the genome, we face about 3 gigabytes of this. And what we really want to know is where the genes are and, you know, those functional chunks on the genome. And this doesn't give us a clue about that. So how do we locate the genes? That's a big question. And CpG islands can be a proxy for helping us how to identify the genes. So what are they, kind of in a lay way definition? Those are regions on the genome that have higher C and G content than other regions. And the other cool part about this is that CpG islands often occur near the beginning of genes, at the promoter regions of genes. So if we figure out how to find islands, that kind of narrows down the search for genes. We have more information about where to look, because otherwise we're really kind of feeling in the dark, looking for the black cat in a dark room. And so how can we define CpG islands? So when we have a long sequence of DNA, many of us would say, oh, well, it's easy. We have 4 nucleotides, and if they each have a probability in 1/4 of occurring, then we expect C plus G content to be about 50%. And that's a very kind of naive kind of thinking, because it doesn't happen like this at all. And so you have regions in the genome where the content of G plus C is different, let's put it this way. And at different places, the concentration, if you wish, the frequency of finding C and G nucleotides varies by quite a bit. So we want to say that CpG islands are those regions on the genome where you have higher frequencies of C and G. And by the way, for the kind of math portion here, you may remember that G pairs with a C on the double helix. This is not what we're talking about here. So don't get misled. We are talking about a C followed by a G on a single strand of DNA. So this is the point of conversation here. So this content is higher in some places, lower in others. And now Robin is going to tell us why this happens from a kind of biochemical perspective. Why does this occur, and why things aren't uniform, as mathematicians very much would like to be.
There we go. Thank you. So in order to explain this oddity, I have to talk about methylation of the Cs. So this is cytosine. It's in its unmethylated form, it looks like this. And the methylated form just has a methyl group, CH3, in that number 5 position. Okay? That's what methylation does, for Cs. Why do we care? Well, it turns out that there is a correlation between methylation of Cs and a lack of expression of genes. Okay? So let's look at what that can mean. Okay, so here's a DNA sequence. And here's one of those CpGs, a C next to a G. And this C is methylated, okay? That state is, or can be, maintained when that cell replicates its DNA and then divides. And when that happens, we're going to take this thing apart, split it apart, break those hydrogen bonds, and then make a new complementary strand of DNA for each of these two templates. So now we've got something that's half methylated on that CpG, and not so methylated up there and not methylated here and not methylated here. And there's an enzyme called maintenance methylase that sees that as its substrate and adds another methyl in the appropriate position over here. Okay. So that can explain how a pattern of gene expression is perpetuated in a tissue type. So if we're talking about skin cells, they're not going to be making hemoglobin, for instance, right? There's no need. And the cells that give rise to red blood cells don't need to be making keratin, you know, the stuff that makes up hair and nails and so on. So the important thing to remember here is that, generally speaking, if there's no methylation on the Cs, those genes are likely to be expressed. And if there is methylation on the Cs, those genes are less likely to be expressed. That's just a general correlation. It's not absolute, but it has effects, as we will see. Okay, now, what we see happening over evolutionary time is this very peculiar loss of certain CpG elements. So if each of these, let me find the, okay, if each of these vertical lines represents a place where a CpG exists, okay, and here's an important gene that does something interesting. We will see methylation of most of these, with the exception of the ones that are just in front of this important gene. Okay? All right. Over many, many years of evolution, what happens is that these guys that tend to disappear. And we're left with these guys who are not methylated. And that's what a CpG island is. But why does that happen? Okay? Why do these guys disappear, right? And these get maintained? And here's the reason. Just as a consequence of existence, nucleotides suffer errors, and in this particular case, deamination can occur. Now let's just go back real quick and notice that this C has an amino group. All right? So this C has an amino group. And a mistake, an error, it's not a mistake, it's a mutation caused by spontaneous chemical reaction, removes that amino group. But the thing is, if you take the amino group off of an ordinary unmethylated cytosine, you get uracil. And uracil is not supposed to be in DNA. It's only supposed to be in RNA. If, on the other hand, you take the amino group off a 5-methyl cytosine, you get thymine. And that is supposed to be in DNA. Okay? It doesn't look as foreign. Repair of that U back to a C is pretty efficient. Oh, it means taking off the amino group. Let me just scoot back. C has an amino group, okay? And just by random chemical action.
That HNN,
yeah,
HNN
yeah,
is an amino
that's an amino group, yes.
group.
No. That's why we're doing this, right? So we learn to speak each other's language. Okay? So this chemical accident of deamination takes this and turns it into a uracil, but takes this and turns it into thymine. Right? Uracil is obviously out of place. Thymine is not so obviously out of place. Whoops. Okay, so repair of U's is much more efficient. Enzymes whose job it is to look for these kinds of errors see them and fix them. So whenever deamination gives rise to a U, it will get fixed back to a C. There's also repair of the thymine back to a C, but that's less efficient. That does not happen as efficiently because thymine just doesn't look as odd as uracil does to the enzymes in the cell that do this thing. So what happens over evolutionary time is that the 5-methyl CpGs mutate to TpGs, and the unmethylated ones are repaired and stay as CpGs. And since we find the unmethylated ones in front of those genes, they tend to be maintained, and that gives us CpG islands. Okay? So there it is. That's the result, CpG islands.
What's the original source of methylation to begin with?
There's another enzyme that does that. So in development, there's this wave of demethylation, and then appropriate methylation occurs, and that's maintained throughout the subsequent mitoses. Yeah.
I'm just curious, in sexual cells,
Mm-hmm.
during passing of genes, does the methylation maintain?
It depends. Yeah, it depends. It really does. It depends on the gene. I mean, there are certain genetic diseases where it matters whether you get the allele from your mother or from your father. Yeah, so yeah, it depends on what genes we're talking about.
But it can happen.
Yes, it can, definitely.
So is there something about the genes that prevents methylation?
Oh, okay, yeah. Generally speaking, if there are methyl groups, then there are changes in. I'm sorry, I'm gonna have to go. I have to go here. DNA in our cells is part of a complex association with protein. And that protein has really important consequences on what's happening with that DNA. So what we see where Cs are methylated, the conformation of that protein DNA association is really tight, such that other proteins can't get at that DNA, right? So, and that's called heterochromatin. It's very dense. You can't get at it. Okay. On the other hand, if the Cs are demethylated, or just regular Cs, the association with the chromatin is different. It can be opened up. It can be made accessible. So that's the short answer. Does that make sense?
I
Okay.
think the bottom line is, if it's a gene that you want to use, you don't want it methylated. If it's one
Yep.
that you want quiet at a particular time or most of the time, you stick on methyl groups.
Yeah.
There are other ways to control this.
Yes. Yeah, absolutely. And again, it's not an absolute correlation, but it is good enough, right? And it certainly results in this change over time. Okay, so one reason that CpG islands are important is that they can help us find genes. I mean, we have other ways of finding genes, but the more tools in the toolbox, the better. Another reason that it's important is that as we have learned more about the changes that give rise to cancer, we used to think that you just had to accumulate a number of mutations in a couple of different kinds of genes. And that's certainly true. We can find mutations in a couple of different kinds of genes. We see mutations that cause overexpression of growth-promoting genes, and we see mutations that cause lack of expression in growth-suppressing genes. And those both happen. But we also now see that inappropriate methylation can have the same effect. If you've got a good growth promoting gene, if you've got a good growth suppressing gene that becomes inappropriately methylated, it's going to be shut off. So the consequences for the cell are the same. It doesn't have the growth suppressor, and that can be another one of the steps that contributes to the development of cancer. So there we are.
Okay, so we have some good reasons to want to know.
Change the battery.
Oh.
Sorry.
Okay. Okay, so how do we go about it? And the naive approach would be, okay, so given a long sequence, I will compute the number of occurrences of A's and C's and T's and G's, compute their frequencies, and here I had a length of 490, and I've computed those frequencies. And if I asked you now, is the sequence part of a CpG island? Or if I asked you, does the sequence contain a CpG island? What would your answer be? You're a fast learner. Well, I mean, we are in a group of mathematicians here, so if you ask me about whether something is there or not, or something has the property or not, I'll ask, how do you define this thing? I mean, what's the definition, right? So what exactly is a CpG island? So we've been all talking about, elevated levels and this and that, but we don't seem to have a definition. And the definition comes, it's a very kind of hands-on experimental definition in the naive approach. And it comes from the fact that if you look at, so here you have a 40,000 bases, and in some places this content of CpG, the rate of CpGs, goes higher than in other places. And so approximately 20%, based on what Robin was saying now, of the genome is C and Gs, and the rest of it is the other two. So that's one thing. I mean, it looks like, above a certain threshold, we have those places that look more likely. And here, those ticks, if you can see, they're the locations of CpGs, and there is some smoothing going on here, so they're not exactly the islands. But you see that there is a cluster here, and then that goes up. And then if we consider this combined percent, one of the definitions is this. We want to have the total content be high, and we also want we also want another characteristic which is based on this observed over expected frequencies to be there. And you have a bunch of kind of commercial products that you give it a sequence, they will give you locations of islands. Just a few of them are listed here. In the package that you'll download with all the materials, there are a few papers that make references to those things, but if you Google them, you'll get right to the site. So, and these are the cutoffs that they use. So content of C and G bigger than 50%, based on the fact that what we would expect is about 20-25%. That's kind of high content. I'm sorry? Well, this slide here for that particular sequences that I showed was about 20%.
So that's not general.
Yeah, I mean, from what I've understood, it doesn't make sense to kind of average over the whole genome that much, but yeah, I mean, it's specifically for that one is what I'm talking about. And this is just the guideline because it's not an exact science, and that's why we'd want to get to some models and kind of make it more mathematical. But the rule of thumb is if you have content that is C and G content combined greater than 50%, that's good. And then you look at things that, so let's look here. So this is near the transcription start site. and see how the CG content goes higher. And there is another characteristic which is called the normalized CpG content, and this is the observed over the expected count. And so that thing also goes higher near those transcription start regions. So based on this, a lot of authors have decided that actually if you want to look at both of these characteristics, so here we're looking at total CNG content, and here we're looking at observed over expected count. And if that observed over expected count is bigger than 0.6, that's probably an indication.
Hold on. If it's observed over expected, wouldn't 1 be the average?
Remember that you are not really comparing with the uniform distribution, so they both go up, right? So, so when you're looking at a sequence, they both go up here. So why would you want it to be 1? I mean, I want it to be something that is kind of experimentally showing an increase, but my baseline is not the kind of uniform distribution.
Oh, so your condition.
So observed over expected is, the observed count is within the sequence that you're looking at.
So
right.
expected is condition.
Yeah.
This is distance from a certain site.
We're talking about if I have here a promoter or start of the so this is a site that we want to focus on because this is where we know we might find those objects.
So we're talking about computing these quantities as a function of
The position, exactly. We want to locate them. Yeah, I didn't pay him to ask this question, but we're getting there. So that's exactly where we're going. But that's the kind of more naive approach. But for this window that we'll be doing, and I'll show you in a second, this is kind of important that you have those two go up, right? And you say if one is bigger than 50% and the other one, the observed over expected, is bigger than 60% or 65%. So there are various papers. The original paper went with 0.5 and 0.55 or 0.6. And then it was finding a lot of repetitive regions that weren't really CpG islands. And then there was another paper by Takai and Jones. The next slide is taking the one figure from there. They kind of made the criterion stronger so that they could eliminate those regions that are of no interest to the question in many cases. So exactly. So once we know that these levels are elevated, you want to do some kind of sliding windows approach to find them. And the sliding windows approach, this is from Takai and Jones. And this is what many of those algorithms still use as the kind of working method, is you take a window of a certain size. And this certain size is based on how big do you want your islands to be. So if you're looking for islands that are bigger than 200 base pairs, you start with a window of 200. If it could vary up and down. So you take that window, you compute the characteristics, and then if you don't meet the characteristic, you advance by one base pair or by a number of base pairs. Different softwares use different approach there, until you find a place where you have a window that fits the criteria. And then as long as it fits, you kind of tile it. And then you see whether the next window also fits the criteria. And if it does, you tile it again. And you keep doing this until you get a window which doesn't fit the criteria. And then you start sliding back. Say, okay, whoops, I went too far. Let's do that. And so you're evaluating all of this. And at some point, you actually have to make sure that the combined thing meets your criteria. And if it doesn't, you kind of chop up sequences from the ends to make it smaller from both ends. And eventually you arrive at something that says, okay, here's a chunk that meets all of those specifications. And if you found chunks that are close to one another by just a little bit, you merge them. Because you say, okay, I'll consider this one big up. And the interesting thing about this is that you would think that this is an exhaustive search. But it's not. So turns out that you can miss quite a few CpG islands going this way. And there is a paper in your package that actually is the paper that published those findings, how this algorithm, although it looks like an exhaustive search in a certain way, it can miss quite a lot of islands. So although this is the kind of working method, the method that a lot of software that is out there and is easily available and web-based and everything uses, you can miss. And so we want something better. But let me show you, in the cases when it works, this is a chunk of chromosome 19, and these are those two characteristics, so CpG and content C+G. And so you see that they kind of go simultaneously up here and here and here and here, and the green chunks are where you think the islands are. And then what you have down here is those green chunks are blown, they're kind of zoomed in to see what happens. And you can see how in certain places the concentration of CpG is high. So it does work for most practical cases. We're just saying, as mathematicians, one can actually ask whether you find all of them, and the answer is no. All right, so what can we do better? We can try to use a model. And the model that I'm going to talk about is based on Markov chains. And so we will go to hidden Markov models for Markov chains. And if you're trying to teach this into a probability class or a math modeling class, you can skip a lot of this introduction. You can go right to what we were just talking about. So as long as your students know that CpG islands are something that has higher content of C and G, and it's important for some reasons, you can really jump to this one. So the model that we're going to use is, imagine that you have this girl whose name is Diane. And Diane has two tetrahedral dyes, and they're differently loaded. And Diane switches between the two dyes using a Markov chain. So she tries not to switch too often between the dyes. So she picks a dye to start with and has a high probability of rolling that dye again. But she also has a probability of switching to a different dye, which is low. And that will make sense, hopefully, in a minute. So this is the kind of mechanism that we will play with. So you have dye 1 is the island dye, and dye n is the non-island dye. And they're loaded differently because the island dye has higher frequencies of C's and G's, and the other one has lower frequencies of C's and G's. And so this sequence that's telling me which dye has been used to get to a certain base on a certain roll of a dye is a Markov chain. So this is the transition matrix of the Markov chain, and this is the Markov property. So I really don't care what happened in the history. I happened where I am now. I care where I currently am, and that determines where I'll go, the probability of where I'll go at the next moment. And just for those who haven't really done Markov chains for a while, Markov chains are really nice because if you have a sequence, you can compute right away the probability of that sequence happening using the transition matrix. And so, for instance, if I start with either this dye or that dye with probability 0.5, and if I want to see what's the probability of generating the sequence I, N, N, I, right? This probability is going to be, okay, the probability to pick the I dye to begin with, then the probability that given I picked the I on the first step, I will still pick it for the second step, and so on and so forth. Those probabilities really can be taken right from the probability matrix, from the transition matrix, and therefore you have the probability. So this is a pretty straightforward computation. Students really understand that right away, so no problem here. Now the problem is that this thing that switches between the dyes is hidden. So Diane doesn't tell us which dye she uses to generate the C, G, T, and what did I miss? A that compose the sequence. So each one of those guys here in the sequence can be generated either by the island or the non-island dye. And we call that sequence the observed sequence. the emitted sequence. So a lot of what I'm going to talk about here was developed for the purposes of speech recognition. So a lot of language is kind of related to emissions of certain symbols or sounds or words. But this sequence that we can see is called the observed sequence. And the sequence that we don't see, the sequence that tells me which dye is used, is called the hidden sequence. And any sequence, any hidden sequence, can generate this sequence here, right? I mean, unless I have some kind of non-island dye that has 0 frequencies for probability for getting one of the 4 letters, and I mean, that really is biologically infeasible. I have really no way of knowing. So if I'm looking at this sequence, I'm saying, you give me any sequence of I's and N's, it could generate that one here. The thing, though, is that they will do it with very different probabilities. Right? So that's the thing, that we want to take this information, take how the dyes are loaded. And this data comes from Durbin. That's analysis of probabilistic methods for analysis of DNA sequences. So you have the reference later in the slide. And so you have actually a distribution of island and non-island frequencies based on some actual data. So this was taken from annotated DNA, and so those numbers are not made up, is what I'm saying. So they come from some real data. And this is how that works. The first dye has those weights for the 4 bases, and the non-island dye has those. And we will be switching in between as a Markov chain with a probability from switching between the dyes kind of relatively low. Yeah.
Just to clarify, we are talking about a Markov process that's moving down the sequence. So if you currently have an A, there's a certain probability of getting a G conditional that you're handling.
No, so at the moment, excellent question. So at the moment, we're really using a Markov property on the hidden Markov chain. And after that, everything for this one is independent. We will get to a dependence toward the end. Because, yeah, I mean, it's a lot more realistic to think that you will have another type of Markov property here on the emitted sequence. Yeah. So, again, this is the sequence. This is the question. So we really, now understanding that any hidden sequence can generate any observed sequence. The question is, how do we find the hidden sequence that is of maximal probability? So we want to know what sequence, so let me back off here just to repeat it. We want to know what hidden sequence pi will generate the emitted sequence X with maximal probability. And because it's difficult to talk about bases and this and that, at this point, we want to do the analysis using really the kind of most common language that you use in probability questions, and this is rolling dice and talking about this out of the context of biology. We will come back to the question in terms of biology a little bit later. So this is again from Durbin, and he refers to this as the occasionally dishonest casino. So those 2 guys are running an operation there that most of the time, so they have 2 dice. So the game is, just roll a die, and you win if you get a certain number, you lose if you don't. But they have 2 dice, and one of the dice is fair, and the other one is loaded. And they are very careful about switching between the dice because they don't want to be caught, but they infrequently would switch from the fair to the loaded and vice versa. And this is how, let's say, this happens. So you don't have to switch between the dice with the same probability. You can really kind of define your transition matrix here for this Markov chain any way you want, but it makes sense for those guys here to be low. And then this one has a really I mean, this one has a uniform distribution. That's the fair die. This one has a really high probability for rolling a 6. And we want to know the most likely path of hidden states that can emit a sequence of numbers from 1 to 5. All right. One more element of notation. So this conditional probability that being in a certain hidden state, I want to see the state B here in the observed sequence, are called emission probabilities. And the emission probabilities really do not depend on anything else than the value here. And in our case, K can be fair or loaded, right? One of those two. So here is how that works. So for instance, EU6 would be, I want to know what the probability is that if I'm using the unfair die, so U for unfair, it should be L for loaded, but U for unfair die, to roll a 6, to see a 6 on the 7th roll. So this is the 7th place here, and I want to know what the probability of emitting a 6 there will be if I have used the unfair die. And then in the same way, what the probability of rolling a 4 on the 10th roll if I'm using the fair die. And that's going to be 1/6. Okay. All right. And just the last bit of notation, these are going to be the transition probabilities for the hidden sequence for the purposes of actually understanding the algorithms, which I'm not going to go much in depth, but just for those who will be reading the chapter that's included, this is going to be used for denoting the transition probabilities for the hidden model. All right. So let's look at some questions. Let's say I know both x and pi, both the known and the hidden sequence. How can we compute the probability that the observed sequence x is generated by the hidden sequence pi? That's now a fairly easy problem. And here is an example. So this is a straightforward problem. So let's say I want to know what the probability of fair, unfair, unfair, using the 3 dice in this order, is to generate the sequence 2, 6, 6. Okay? And this actually means, okay, let's start with picking the die at random. So that's a probability of 0.5 that I'll pick the fair die. And then I want to multiply this by the probability that the fair die will emit a 2. And then I want to multiply this by the probability of switching from F to U, which is the conditional probability of the Markov chain. And then I want this U to emit a 6, the unfair die to roll a 6. And then I want to switch, which is in this case not switching. I want to stay with U, which happens with probability A, U, U. And then have that last U emit a 6. And when you multiply those probabilities, you get this. And then you say, okay, well, let's see whether there's a difference if I want to generate the same observable sequence but using a different hidden sequence. And you use the same thing using the appropriate transition and emission probabilities here, and you get a different probability by an order of magnitude. And those could vary by quite a bit. All right, so why do I want to know this? Because I eventually would like to know what the probability of the sequence x is, just by itself, because I don't know pi. This is hidden. And the answer, if you ask your probability students, of course, that's a very good place to talk about, we can apply the total probability formula. We can sum over all of those hidden sequences, right? We have a sequence of a certain length, and we have only 2 states for the hidden sequence, finite in any case. So we have finitely many sequences. And we can sum over all of those sequences. We know how to compute the probability px pi for any x and any pi. And after we sum up over all pi's, we'll get f of x. And of course, I chose a very short sequence. So here is an illustration. We can compute this, no problem. What your sequences don't know, and I know, of course, everybody knows here where I'm going, is that this is not biologically feasible because with n equals 90, and so a 90 base pair sequence is nothing from the point of view of looking at DNA sequences. You get to a number of hidden sequences that's of this order of magnitude, and this is about the number of atoms in the observable universe. And so actually my students quite enjoy doing a kind of by hand computation when I say, okay, how many operations, flops, do you think your computer can do in a second? And they go this many. And I say, okay, I'll give you 10 to the 5th more. So let's think that this is a fast computer, super fast. And so we go down the path of computing, how long it's going to take, and by the time we have really kind of used, okay, I'll give you 10 to the 3rd more, or 10 to the 10th. I'm really generous today. I feel good. We kind of come up with answers that are 10 to a kind of insane number of years that will take to compute this. So each one of us knows why this happens. The interesting thing is every time I've done this with students, they are surprised. Because everywhere we turn these days, we hear how fast our computers are, and we can do anything. We are so great. Not there yet. So brute force still doesn't work. And of course, this is just for 90. We want to do thousands here, right? So just forget about it. All right. So what do? Let's just state the problem once again. This is what we want to do. We want to look at X. That's going to be my input. And I want to find a path that maximizes this probability. And of course, it's a nice question to see that the maximum number of paths, because you have multiples there with the same probability. The interesting question here is that, of course, the maximum of this also gives you the maximum of the conditional probability. So the conditional probability of X given pi. All right, so that takes us to dynamic programming. Matt may mention something this afternoon, and I'm not going to go into details here. So this is a point where, if you've taken your students to this place, you can say, okay, that will be a nice project, nice research project, depending on how far you want them to go with this. So Viterbi's algorithm basically is based on that. Okay, I'm going to do it dynamically, and each time I'm going to look at the most likely sequence that will end in a certain place. So for instance, I can say, if I want to compute the most likely path, I'll say, I will look at this, and I want to compute the probability that is most likely that ending at L in this place will emit a 4. And then you will compute this probability based on the probability of the maximal path that took you to F here, and then consider the two options, whether that F has emitted and then consider the two options, whether this one is emitted by the F or by the L, and take the maximum. So each time I'm computing something, and then I'm taking the maximum, and that can be expressed this way. So let's see why. If I want to compute the probability of the most likely path that ends here in a state M, where M can be loaded or fair, with an observed value this, I'll say, well, I have a couple of ways of getting there. I can, in the previous state, I can have the maximal sequence be either in a loaded or fair state. So it could be either L or F. And then if I have done this, then I have to transition from K to M to be in state M with at time I+1, or for the I+1 entry of this sequence. So this is, I'm taking the most likely probability that has taken me there, and I'm transitioning to the other state. And then, of course, I have to make sure that this M state emits the symbol that I want. Okay. And I will have the larger of those two taken, where K can be loaded or unloaded. And so here is a kind of more numerical example. So if we dispense with the i's and I plus 1s and so on. So let's say I want to compute the most likely sequence that, that will give me a 1 here from a hidden state L. And you will say, OK, that actually means that I could be at x4, at this 5 here, from a loaded or a fair sequence. And if I happen to be at the loaded, ending in place 4 with the loaded, I have to stay with the loaded die. And if I happen to have used the fair die to get the maximal probability sequence, I'll have to switch. And in both cases, I need to emit the 5th element here, which would be the 1 in this case. And I want to take the maximal of those 2 options, which will give me the maximal probability overall to do what I want to do, to have the maximal sequence, that maximal probability sequence that ends at L, emitting the symbol 1 in my case, x5. So again, this is just for us here to talk about. If you want to do this in a more kind of strict way, you have to ask a couple of questions. How do I know this is really the maximum likelihood sequence overall? And there is some theory here, and it helps in the chapter that you will download with the materials. You have a numerical example that really takes a short sequence and applies by hand Viterbi. And after you've done this example, you will know why you never want to do this by hand. But it kind of walks you through, and of course, implementing this algorithm, you need to have some pointers that track you backwards when you've gotten to the point you need to trace back the most likely sequence. But this is very much better. So it's quadratic in one. I think it's quadratic with respect to the length of the sequence and linear with respect to the number of states, or the other way around. But it's a lot better than the exponential growth. OK, so in that sense, when we're talking about the occasionally dishonest casino, we are trying to identify the U islands, right? So let's say I want to know where those guys are cheating on me. When are they using the loaded die, the unfair die? And so I may be looking at this and saying, well, gee, there are some places, maybe here, I have 2 6s in a row and then another 2 6s in a row, so I'm getting suspicious because 6 is appearing more frequently. But of course, it's all probabilistic, so anything can happen with a different probability. And so if I want to do this, I want to identify the islands as the crossings from the fair to unfair die. So I can say, all right, when I look at the hidden sequence, and the hidden sequence is U's and F's, where I cross from fair to unfair, this looks like an island to me. And here perhaps that looks like an island. So this is what the Viterbi algorithm does to do the initial decoding problem. And then you don't want things to switch too often. So if 2 such switches are close to one another, you can kind of put them together and say, OK, these are not 2 tiny islands. This is actually 1 bigger island, and so on. And there is some decisions. I mean, you have to decide when to do this and what the cutoffs are and whatnot. Now, when I mentioned the other day that good collaborations come out from forums like this, I meant this example because I was giving a similar talk a couple of years ago, and I was saying, I know how to calculate those things, but I'm patching up different pieces of software and moving things from one software to another, and it's just not nice. And the thing that I wanted most of all was, OK, this problem about CpG islands, we don't know where they are, so it's hard when you apply your method, it's hard to see whether it's doing well or not well. So we actually have to take annotated sequences, and it kind of gets complicated. So I was thinking, OK, can we design software that will simulate things, keep track of what die has been used for the simulations, whether fair or unfair, and then try to run Viterbi and the other algorithms that I'm going to show on that simulated sequence and see how well it does. And so, the person who wrote this software basically said, oh, no problem, we can work on that. And by the way, when you go, so you can Google CPDEducate, and that's the first thing that comes up because there's nothing else that's called this. So Google CPDEducate, and you have a bunch of options there. So the options are, this is the home. There is a sliding windows animation, just if one wants to see how sliding windows work. You can just cut and paste a sequence in FASTA format there if you want. It works pretty slow, and frankly, I don't know whether it works correctly because eventually we decided that this is not where we wanted to go with that. So the other tabs are the dishonest casino, where you have the 2 dice and the dishonest casino problem that I just described, and you can change all of those here. And then you have one that really does CpG island identification. And the cool thing is that you can generate a sequence. So here I've generated, if you go under generation, you can generate a sequence of any length. There is really no limit, so you can generate large sequences. And then you can go to the evaluation tab, and you can paste this generated sequence there and kind of try to decode it. And if you also paste, I forgot to tell you that if you go to this one, in addition to the generated sequence here, so it continues, and you will see the generated sequence here, but you will also see a sequence of zeros and ones that tells you what die was used to generate which entry. And then you can paste that sequence back into the decoding tab, and if you paste both sequences, the ones that were used for the generation are the ones that are highlighted in gray. So the red ones here are the entries that are decoded for applying Viterbi for this sequence. And then the kind of gray shading of places is where the unfair die was used. So the more overlap between red and gray we have, the better the algorithm is doing. And I don't know what you think. I mean, we've missed a bunch here. We've missed a bunch here. But overall, I thought this is pretty darn good. So it was kind of interesting to see this. And of course, if you vary the probabilities, and those algorithms work well when the transition probabilities between the loaded and the fair die are relatively small. Because of course, if you're switching with probability 1/2, it's kind of purely random. So that model is not going to do much for you.
Quick question. I'm looking
Yeah.
in the middle, sort of like 4 lines below that middle strip of red.
Mm-hmm.
And I see, right about
Bunch
there,
of sixes, yeah?
There are 7 out of 9 sixes. And so are you saying that?
Happens. You may win the lottery.
Well, I agree, but I would
Yeah.
expect that the algorithm would at least give that a false
Yeah.
positive. So really-
Well, at the moment we are just saying the algorithm finds the maximal probability sequence, so it doesn't really look at subsequences. So this is global, right? And so that's what the outcome is. And actually, one of the disadvantages of Viterbi is that in certain cases it will miss small islands. That has been reported, and it's
seems
known to
right
happen.
That island that missed that small island, that also was a false island to begin with. It seems a little...
Here, what we are doing is we are saying we're finding a maximal probability sequence. By the way, the probabilities are computed as logarithms because they get pretty small pretty quickly. So you have a pretty negative logarithm here, which gives you the maximal probability for this thing to happen. Okay, so another question, another neat way to decode things, especially when you're doing switching between 2 sites, is the forward-backward algorithm, which is another dynamic programming algorithm, which I'm not going into, but just what does it do, and I'll show you an output, is find a probability that the dealer was using the unfair die at any given time I. So the probabilities here that we compute here are different. Given the full observed sequence, so if I'm looking at a whole sequence, I kind of pick a spot and I say here, I want to know, given that whole information, I want to know what the probability is that I've used the fair die to generate this one. Or the unfair die in this case, the unfair die. Right? And And you can calculate another dynamic programming method. We can talk more about this if you want on Friday. The derivations of those are in the chapter that you'll download. But the neat thing about this is that you can use the software. And so here, we have, again, simulated a sequence, generated a sequence. We have kept track of what state is used for what. And then you are plotting here, so the software will give you this output. And what you're plotting here is, given the whole sequence, give me the probability that at each of those positions I have used the unfair die for the generation. And the gray bars are where the unfair die is used. And of course, on this sequence, you see that you have smaller chunks where you've used unfair die, and they do not show because of the scale. So that happens quite often. But by and large, you're looking at those big peaks for longer chunks of the sequence. And you have a pretty good overlap. Again, not perfect because of the probabilistic nature of things, but a pretty good overlap. And this is under the CpG format. This is under the evaluate tab. So we're evaluating this conditional probability. And then finally, the question of, well, gee, we talked about changing the parameters of those, and how do we, and looking at this model that says we will stay with that die with this probability and transition with another probability. How do we know that? And so this is the same question for DNA sequences, and that's where you train your algorithm. So this is estimating the parameters from real data. So you get annotated sequence, and you will say, all right, assuming this model, assuming that this sequence was generated using this kind of hopping that Diane is doing, and using the dies in this fashion, how do I estimate the parameters? And this is, again, I mean, you can do this. We can talk about you can estimate both the transition probabilities and the emission probabilities. So you really estimating all the parameters of the model from data. And of course, you're counting on having a certain number of annotated sequences or a certain number of sequences where you know how the dishonest casino did its work. Right? So you have a certain number of sequences for which you know both the X's and the pi sequences, the observed and the hidden. And this is just a quick run of the algorithm. So here, of course, what we've done, being able to first generate and then pretend that we didn't know how this was generated, try to recover the parameters, we generated a sequence that had this set of parameters. And that's the sequence. And then when you train it, this is what comes back. And of course, the longer the length of the sequence, the better it is. In this case, we're generating a single sequence, and we're training on a single sequence. Once you get to, I'm using multiple sequences, there is some smoothing that needs to happen, which again, you will find some references to in the resources. Yeah?
Is the training with maximum likelihood?
By and large, yes, actually, yeah. There are some kind of additional things, but yeah, by and large, yes. And one thing, of course, that immediately becomes clear is that if we're training on a single sequence, we have absolutely no way to estimate the initial conditions, because for that you need multiple sequences. So those will always be kind of off by a lot, because it estimates the probability that you started with a certain state. And yes, indeed, you started with that state. But for all the other parameters, that illustrates the method pretty well. Okay, so then, of course, you want to say, okay, for now, I only use the Markov property on the hidden sequence, and I want actually to use it on the actual observed sequence as well. And you can pretend that the A's and C's and G's and T are chosen independently from one another, but that's not the case. The nucleotides matter, so there is certain kind of dependency there between how things behave with respect to their nearest neighbors on the sequence. And so here you can do a couple of things. So you can say these are the distributions of the D nucleotides. And so A followed by A followed by a C, and so on. And that would be the island. And those will be the non-island. And again, you can estimate those from sequences that we know very well, that have been annotated. And then you can go to the tab that really does CpG island identification. And now if you look at this, and I'll let you play with it for the next 90 minutes. If you look at this, that's a lot of parameters to enter. So if you really want to change things, that's a lot of things to change. So as part of this, in the package you will find some files that you can upload. So instead of really entering this every single time, you can save things in a file and then upload it. The file has to be a comma-separated value format. So you can use Excel to create it, but then you have to save it as comma-separated value file. And then you can play exactly the same game here as with the dishonest casino. The values that came, that are the initial values here, the default values for this part of the software, came from that paper, where they did some extra work after initially estimating parameters, and they decided that based on whatever criteria they were using, that would be the best set of values to use. But those can change, and you can use file upload. And so for those of you, I'll stop here and we can talk more. But if anyone would like to know what happens here, so why the algorithms work the way they work and what's behind them, tomorrow I'd like to do my lecture on level 2 for Boolean models and bistability. But then when we do the 90-minute lab, anyone who has questions about those, they are in the chapter, so the careful derivations of those, with the exception perhaps of the details here. I mean, we've kind of glossed over some of the technicalities of the training algorithm, but by and large, a lot is available. So if you look at those, if they're of interest to you, and you have questions, we can talk about those during the lab slot tomorrow. And finally, one thing that I want to say before you ask me questions on this, if you have any, before I forget, is tonight the dinner is here. And we want to have dinner, and that will be really nice. But then I and Terrell and Matt will go to the hospitality suite in the hotel, and we will be there for a while. So if you want to ask questions about anything, literally, if you've tried some of the exercises and have questions, or if you just want to give us some feedback, or we can talk about things that we have done in the classroom and what worked and what didn't. We will have that discussion about the classroom Friday, last thing of the tutorial, but we can, nobody says that, we cannot talk about this until then. So just keep this in mind. If you want to stop by and chat, that's great. And if you feel just so beat from a long day today, which it is, it's fine if you just decide that you've had enough for the day. Okay, so any questions about any of this? Yeah?